Hi everyone, this is the ninth lecture for your chapter Cell. In the last class, we discussed the Golgi complex, which is the second part of endomembrane system. Okay, after endoplasmic reticulum, you have Golgi complex, which is like the postman of the cell. Okay, it is like the postman or the post office of the cell. It sorts the different proteins and different components into separate vesicles. Okay, and it is also involved in production of said lysosomes, okay, which we discussed in the last portion of the last class. Okay, so today we will start with these lysosomes, which are the third members of endomembrane system, and later on we will also be discussing vacuoles. So the four parts of the endomembrane system are endoplasmic reticulum, Golgi complex, um, lysosomes, and vacuoles. Right. So let us begin. Lysosomes, they are also known as suicide bags of the cells. As you can see from the cartoon, what their function is basically intracellular digestion. Now, know the difference between intra and inter. Inter means between two things, and intra means within. So, within the cell means intracellular. So digestion within the cell is carried out by lysosomes. As you can see here, the green colored one is bacteria and the red colored one is the lysosome. This lysosome will come, and once the bacteria enters the lysosome, it starts getting degraded. Okay, starts getting digested with help of enzymes. So that is the function, the one line function. If somebody asks you what is a lysosome, then it is the vesicle, or it is the organelle which helps in intracellular digestion. How does it get its name? So in Greek, lysis means to digest or to break. Okay, you will have heard of this term lysis a lot in biology. So it is important to remember what it means. Okay, so in Greek, lysis means to break or to digest something, and soma means body. So these are small bodies which can digest or break something. Fine. They are small. How small? 0.2 to 0.8 micron in diameter. So if this is a lysosome and you calculate the diameter, it would be 0.2 to 0.8 micron. Okay, so they vary in size. They are single membrane bound vesicle. Okay, they have only one membrane. Now, a lot of students may sometimes confuse between a single membrane and lipid bilayer. See, single membrane means it will still have the lipid bilayer. Okay, it does not mean only one layer of lipids. Fine. Single membrane means it has only one lipid bilayer. Okay, not many. Fine. So single membrane means one lipid bilayer. So it is single membrane bound vesicle containing hydrolytic enzymes in form of granules. So what we see inside is hydrolytic enzyme picture. Now what are hydrolytic enzymes? So from the name itself, now you know the meaning of lytic. Lytic means to break. Hydro means it comes from water. Okay. So hydrolytic means the enzymes which break something, break the molecule, break the substrate with addition of one molecule of water. Okay. So we will study later on, if we study biomolecules, that when you have to break any polysaccharide or polypeptide or any polymer, to break the bonds you need to add one molecule of water. Okay, and then you can break it into its monomers or components, right? So such enzymes are called hydrolytic enzymes. Okay, they are present in form of such granules. The enzyme acid hydrolases. Okay, now inside the lysosomes they are called acid hydrolases. Again, hydrolase means the same thing. Okay, you will study 6 different classes of enzymes. So hydrolase is one of them. Again, it means the same as hydrolytic enzymes. Okay, and what does this acid mean? So acid hydrolase means these enzymes will act. These enzymes will get activated only when the pH is acidic, around 4 or 5. Okay, so the enzymes inside the lysosome are acidic in nature. Okay, they get acid activated only when the surrounding becomes acidic. These enzymes will be able to do their function. Okay, and for this, the membrane of the lysosome has an active hydrogen ion pump. Okay, I have forgotten the word pump here, do not worry. So when you talk about this membrane, it will have proteins as well, right? So in these proteins there are some hydrogen pumps also. What these hydrogen ion pumps or proton pumps do is they take H plus ions, okay, protons from the cytoplasm and concentrate it within the lysosome. When that happens, the hydrolytic enzymes or the acid hydrolases of lysosome become activated, and then they will start digestion. Okay, so this is sort of a safety mechanism that the enzymes should not get activated anytime, anywhere. Okay, only when there is acidification of the insides that the enzymes will get activated. Okay, so this is one safety mechanism. Example, acid phosphatases, meaning the enzymes which will remove phosphate, okay, phosphate group only when the pH is acidic. So such enzyme is called acid phosphatase. Sulfatase, protease, peptidase, nuclease, lipase, carbohydrase. A lot of enzymes are present inside the lysosome. Fine. The membrane keeps enzymes away from the cellular contents. Okay, this membrane, it is actually protecting the cell by keeping these enzymes away. Okay, if by some reason this membrane breaks or there is some damage on these membranes, the cellular contents will start getting digested. Okay, because these enzymes, they will leak out. If the enzymes leak out, they start digesting the organelles or the food storage material or anything which is present inside your cytoplasm. So the cellular contents would be digested, and so they are called suicide bags. Okay, you only break the membrane if you want the cell to die. Now this membrane can become fragile in absence of oxygen. So if somebody has hypoxia, hypo means less, oxia means oxygen. If somebody has a condition like hypoxia, okay, maybe you are drowning, or maybe because of any reason, okay, any syndrome, if you have hypoxia, the membrane becomes sensitive, okay, it becomes unstable, and there are chances that it may break. So the membrane becomes fragile, okay, weak in absence of oxygen. Then in excess of vitamin A and E. When you will study vitamins, you should know that the lipid soluble vitamins, which is K, E, D and A, these vitamins should never be taken in excess. Okay, they have a limit. If you take a lot of vitamin A or a lot of vitamin E, it causes toxicity. Okay, so this is what happens when you take excess of vitamin A or E. It makes these membrane fragile. Okay, then bile salts are secreted by your liver. Asbestos particle, people working in factories or where asbestos is being used, they are prone to um, um, getting in touch with such asbestos particle, okay, through respiration or through skin. Then if there is a lot of heat for some reason, then many drugs can make the membrane fragile. Then x-rays, UV rays, there can be a lot of reason. Even smoking is considered one of them. Okay, so why smoking damages is because the damage can directly be seen on these lysosome membrane, okay? And if the lysosome membrane becomes fragile, okay, if it breaks, the enzymes get released into the cytoplasm and they will do what they do best, which is to digest everything, okay? Now, how are these lysosomes formed? So, in bits and pieces, we have already learned this, that they are formed jointly by the endoplasmic reticulum and the Golgi complex, right? So the precursor of the enzymes are synthesized in endoplasmic reticulum because endoplasmic reticulum has the ribosomes, while it is changed into its final form in the Golgi complex. Okay, a lot of these things you will see the proteins are synthesized in endoplasmic reticulum, but they are modified and they are activated or packaged in the Golgi complex. So the same thing happens for the lysosomes as well. In animals, lysosomes occur in all cell except RBC. Okay, you will see this RBC as an exception for many organelles. Okay, so lysosomes are absent in RBC. They are abundant in cell performing phagocytic activity like WBC. Your white blood cells, especially macrophages or monocytes, both of these, they do phagocytosis. So the cells which are performing phagocytosis tend to have a lot of lysosomes. Okay. So as you can see here, this is what your normal WBC should work like. It takes a bacterium. If you have bacteria in your system, it will engulf it. To engulf means to phagocytose. Okay. Then after getting engulfed, it is fused with lysosome, and this lysosome will digest whatever is there. So if it is a bacteria, that bacteria is digested, and whatever excess residue is left over, okay, that is all thrown out. So this is the function of your WBC or macrophages and monocytes specifically. Lysosomes take part in intracellular digestion of various types of materials, as I mentioned earlier. Depending upon their morphology matlab structures. Okay, depending on their structure, depending on where they are found, how they look like, and their function. So depending on their structure and function, there are 4 types of lysosomes. Okay, there are 4 types of lysosomes. It is easy to remember. You will not have any problem. Okay, do not worry, just understand it clearly. So you have primary lysosomes. Primary means the first. Okay, so the first type of lysosomes or primary lysosomes are the newly pinched off vesicles from Golgi apparatus. If this is your Golgi apparatus or your Golgi complex, from this, when the new vesicles they pinch off, okay, so this first vesicle coming directly from Golgi apparatus is called primary lysosome. It has hydrolytic enzymes present in form of granules. Simple. Primary lysosome coming directly from your Golgi apparatus. It is all new. Okay, hence primary. Then comes secondary lysosomes, which are also called digestive vacuoles. Okay, so they are formed when the phagosome. Now again, just focus here on the diagram. You have a bacteria, and this bacteria is being taken in by receptor mediated endocytosis or phagocytosis. Okay, just consider it to be a phagocytosis right now. So this bacteria is sensed by the receptors on the cell membrane, and then the cell membrane starts to form pseudopodia. These pseudopodia will fuse, and the bacteria would have entered in this small what you can say sphere, which is now called a phagosome. Wherever you hear the word phag, phag, okay, or phage, it depends on how it is used. You can pronounce it in both ways. It means food, okay, or something to do with eating. So phagosome, which has this single layer, okay, a membrane formed from the plasma membrane. It has the bacteria or the food particle inside. Now this phagosome will fuse with the lysosome. Okay, remember the primary lysosome we just talked about? So these primary lysosomes will fuse with the phagosome, and when that happens, all the enzymes from lysosome will start digesting the uh, bacteria or whatever food particle is present in the phagosome. Okay, so this fused vesicle or the fused organelle is called phagolysosome. Clear? So fusion with food containing phagosome, digestion occurs, then absorption. Okay, after digestion, whatever sugars, amino acids, vitamins, whatever important things are there, they will directly get absorbed into the cytosol, okay, or the cytoplasm, and the rest undigested food will be thrown out of the body, okay, out of the cell here. So then comes tertiary lysosomes. Tertiary means third, okay, third type. They are also known as residual bodies. Now what we just discussed, the undigested particles, okay, which cannot be digested further, these should be thrown out, okay, they should be thrown out by exocytosis. So this vesicle will move and it should fuse with the plasma membrane, and that is how the contents should exit the cell. If okay, so this happens by exocytosis or apophagy, which means the same. If for some reason these residual bodies, okay, which have this undigested food, if they are not able to fuse with the plasma membrane, or if exocytosis is not happening and they remain within the cells, scientists have found out that it leads to aging. Okay, so aging can be caused because of large number of residual bodies in the cell. Okay, tertiary lysosomes, residual bodies, they are leftovers of the phagolysosomes, and they have material which cannot be digested further. Okay, it is supposed to be thrown out. And finally, the fourth and sort of interesting one. Okay, so pay attention. The fourth one is called autophagic vacuoles or autophagosomes. Now, from the term itself, let us try to understand. Auto means self. Okay, how you have autopilot means it will self drive itself, right? So, auto means self, phago means to eat. So these lysosomes, they eat the whole cell. Okay, so the cell is eating itself with help of lysosomes. So it is called autophagosome. Or the same thing is applicable in this term also, autophagic vacuoles. Autophagy again means to eat yourself. Okay, so the cells here are eaten themselves or the cell organelles. Fine, let us understand that. For digestion of worn out organelles. So after some time, after the organelles have already been formed and they have been working, there is a possibility that they become worn out. Okay, they have some damage in them, the organelles, or they are not functioning properly, they are not efficiently working. Okay, the efficiency has decreased. In such cases, these organelles are digested to be eaten. Okay, and this phenomena is called autophagy or autodigestion. And so lysosomes are also called disposal bags to dispose of, right? Just see here, you can see there is an autophagosome forming around the mitochondria, which are supposed to be digested because they have become old. This autophagosome will it will fuse with the lysosome, and when it fuses with the lysosome, the hydrolytic enzymes within the lysosome will start eating away at the organelles, and the organelles will be digested. Okay, whatever important materials will be there can be absorbed back into the cytoplasm, and this will become again a residual vacuole or residual vesicle, which will do exocytosis, and the cell organelle debris, whatever cannot be digested, the undigested material will be thrown out of the cell. So this is what autophagy means. The part which is not functioning properly, which is damaged, will be eaten. Okay. Then there is a term called autolysis. Again, same thing, self. Lysis means to break. This is also known as apoptosis or programmed cell death. Ever wondered like who decides whether the cell should live for let's say 10 years, 8 years, 5 years or more or less? So actually the cell itself can decide when it becomes old and it is not able to function properly. It does this self destruct. Okay, have you seen in some movies they show there is a self destruct button? Okay, self destruct meaning to destroy itself. So this self destruction is called autolysis, apoptosis, or programmed cell death. It happens when there are old diseased cells. Okay, either they are old or they have disease, some sort of disease, cells, tissues, or organs. Okay, this is also used when there is some death, cell death suddenly in your neighborhood. If uh, there have been a lot of cell deaths, to clear that cell debris away, you also require lysosomes. Okay, so the disappearance of larval organs during metamorphosis is also due to autolysis. Okay, not always it is destructive. Sometimes it is also constructive. Like we will see in the next slide what it means. So metamorphosis, have you heard of this term? To morph means to change from one structure or one shape to another shape. I'll give you example of metamorphosis. Think of a butterfly. Okay, it is firstly a caterpillar. That caterpillar becomes a pupa, and from that pupa comes a butterfly. Okay, so this whole process is called metamorphosis. Same is seen with frogs. Frogs are in starting they when they hatch from the eggs, they are actually in form of tadpoles. These tadpoles have tails, okay, but the adults do not have. So this metamorphosis requires the tail to be destroyed, okay, to go away. So what happens is there is autolysis in the cells of the tail. Okay, this autolysis will destroy the cells of the tail, and whatever important material will be released. Okay, whatever nutritious material will be absorbed back into the body, and the cells of the tail will be degraded away slowly, and finally the adult frog will not have any tail. Same is the case for us. If you look at the human embryo, okay, and how it progresses, you will notice that this is how the fingers are formed. So this is your hand before, and between these regions you have this small dark colored bands. These bands are actually showing where the apoptosis is taking place. Apoptosis or autolysis. Okay, so cells here will die. Okay, they will be lysed, and only after their lyse will the 5 digits, the 5 fingers, separate from each other. Okay, so this is the adult hand. This was possible because in your when you are a human embryo, okay, there was such kind of autolysis which helped in formation of 5 fingers. Clear? So lysosomes, autolysis is to save you, to protect you from the dead cells or from the cells which are not functioning properly, who have become old, who are diseased. So to protect you from such cells, okay, which can harm. Second thing, as I told you, apart from destructive, it also has some constructive side, which we just saw in organ sculpting, or where the during the metamorphosis, some of the um, organs or some of the part of the organ are destroyed, okay, to give rise to this adult form. Fine. So here is an overview of what we just studied. Okay, a very quick recap. You have Golgi complex. From Golgi complex, you can see there are enzymes which are concentrated here. So if these enzymes are the precursor of hydrolytic enzymes coming from endoplasmic reticulum, they are made what you can say, they are made into active and proper folded enzymes in the Golgi complex. Then they slowly start to pinch away, okay, and as that pinching happens, they form this primary lysosomes, okay. Primary lysosomes can be used for two functions. It can be used for intracellular digestion, as well as sometimes when I told you that if you have a lot of dead cells in your neighborhood, what you should do is, or what the cell does is, it secretes these hydrolytic enzymes outside, okay. So this primary lysosomes, they can fuse with the plasma membrane by exocytosis, and these hydrolytic enzymes will be released outside, and this helps in clearing away the dead or debris of the cell. Fine. Now these primary lysosomes, they have the enzymes, okay. Now let us say you have phagocytosed bacterium. So here can you see this blue colored bacterium? It is taken inside. This is called a phagosome. This phagosome will fuse with the primary lysosome. When that happens, it forms a heterophagic lysosome or secondary lysosome. This lysosome will finally digest your food, okay. After your food is digested, all the undigested particles, okay, and whatever is nutritious will be absorbed in the cytoplasm. The undigested particles remaining within the phagosome will now form the residual body, which are the third type of lysosomes, okay, tertiary lysosomes. This residual body, by exocytosis, all of the undigested material is thrown out of the cell. Also, when some of the organelles are not working properly, what happens is your ER, okay, endoplasmic reticulum, it forms such kind of a what you can say, a jail around the mitochondria till the lysosome can reach it, okay. So it will form such kind of covering, and when these autophagic vesicles, they fuse with the lysosomes, the organelle starts to get digested, or it starts getting degraded, and again the same thing happens. Nutritious things will get absorbed into the cytoplasm. The rest will be called residual body, which should be exocytosed, okay. And the last part is when there is autolysis, okay. So the primary lysosomes will be ruptured, okay. They will rupture inside the cell. The hydrolytic enzymes will be released in the cell, and they will start digesting anything and everything they come in contact with, and such, so the cell will finally die, okay. So this is autolysis. Intracellular digestion, these are the functions of lysosomes. A quick recap: intracellular digestion, extracellular digestion, which I showed you in the last diagram. The lysosomes release enzymes in the external environment through exocytosis. Body defense, the leukocytes or WBCs, okay, they eat bacteria, devour bacteria and other microorganisms, and these bacteria and microorganisms are digested by with help of lysosomes, okay. They are made non-active or non-harmful by digesting them with lysosomes, okay. So thus they take part in natural defense mechanism of the body. Then autophagy. Autophagy is important during metamorphosis of many animals, okay. Certain embryonic parts like tails, gills, etc. are digested through lysosomes, and this whatever digested material will be there, the nutritions will be used for growth of other parts of the cell. Then removal of obstruction. Sometimes if there is any obstruction for the cell growth or cell development, then what the cell does is it exocytosis happens with of lysosomes. So the enzymes will be released outside, and the obstructing structure, whatever is obstructing, can be removed, okay. The obstruction can be removed. Mobilization of reserves. So if uh, there is starvation meaning you are not getting enough food, so lysosomes provide nourishment by rapidly hydrolyzing the food which is stored in the cells, okay. The carbohydrates, fats or proteins in whatever small amount, if they are stored in the cell, these carbohydrate, fats and proteins are made available to the cytoplasm, to the cell, okay, for survival. Then mobilization of reserve food during germination of seed is also accomplished by lysosomes. If you know a seed, okay, it has a lot of stored food. So this stored food in form of complex polysaccharides or polypeptides is finally mobilized, okay, made into simpler molecules and made mobile. Mobile means movable from one place to another in germinating seeds. Intracellular scavenging. Scavenging means to make sure that the old and less efficient organelles are removed. Okay, so this scavenging, intracellular scavenging is also done by lysosomes. Then disposal of useless cells. They can break down aging and dead cells. Formation of hormone thyroxine is also, I mean, so when thyroxine or T3, T4 hormone, okay, which is synthesized in your thyroid gland. So during its synthesis, there is a protein called thyroglobulin on which these modifications will occur and the thyroxine molecules will be synthesized. So to separate the thyroxine from the thyroglobulin protein, lysosomes are required. Okay, so that is why it is said formation of hormone thyroxine. If you can remember this, good. If not, it's okay. Right? So there we end with the lysosomes, which were the third part of endomembrane system, and we move on to the last, which is vacuoles. Vacuoles are again single membrane covered bags in cytoplasm. They are believed to be formed by expansion and pinching off from endoplasmic reticulum. Okay. Depending on the contents and function, vacuoles can be of 4 types. You can have sap vacuoles, contractile vacuole, food vacuole, or air vacuole. Okay, 4 types. Now in this diagram, this is a electron micrograph. Okay, it is an electron micrograph, and you can see whatever the empty space is seen in between, that is actually not empty. It is the large vacuole, okay, sap vacuole that you see in plants. These other organelles you are seeing are the chloroplast, okay, and then you will have the nucleus also. So this is a typical plant cell, and you can see a vacuole takes up a lot of plant space in the plant cell. So we start with the first type, which is the sap vacuoles. Sap means fluid, okay, it is a sort of fluid. So sap vacuoles are fluid-filled vacuoles having selectively permeable membrane called tonoplast. These vacuoles have a membrane, okay, a single membrane, but that membrane will have a lot of different channel and carrier proteins, which will allow only specific things to enter the vacuole. Okay, so they are selectively permeable membrane, and that membrane is called tonoplast. Okay, this vacuolar membrane. The fluid is called sap or vacuolar sap. In mature plant cell, a large single vacuole occupies 90% of the space. Okay, so when you look at the plant cells, you can find these large molecules, or sorry, not molecules, large organelles or large spaces which seem empty. So they are nothing but they are vacuoles. Now, what do these sap vacuoles do? So they may store food like sucrose, then different mineral ions, amino acids, esters, proteins, but they also store waste products. Okay, so basically it is like a what you can say, a storage. Okay, a storage house where you can put your waste also, and you can also put whatever you might require in future, like sugars and proteins and other food reserves. Okay, so the food is also stored there, the waste is also stored there. And water soluble pigments like anthocyanin, you know, some of those vegetables and flowers, they have very nice red color. Let me give you an example of beetroot. Have you eaten beetroot? So this beetroot is red in color, right? It even stains your fingers. So the beetroot is red because of presence of this water soluble pigment. Pigment means some colored material called anthocyanin. Fine. So anthocyanin. Then you must have also heard of purple cabbage. Have you seen purple cabbage? So that purple cabbage is also because of anthocyanin. Clear? So this anthocyanin type of soluble pigments are also present inside the vacuole. Okay, sap vacuole. And finally, the main part is they maintain osmotic pressure in cell, and it maintains the cell's turgidity. Okay, remember when we discussed endoosmosis, exoosmosis, plasmolysis. When plasmolysis happens, meaning outside the cell there is hypertonic solution, so the water will start moving outside. From cytoplasm the water will move out. First the water moves out from cytoplasm, and then the vacuole will also start shrinking. The water from vacuole will also start going out of the cell, and then you call it incipient plasmolysis. If you have read about it, good. If not, when we discuss transport in plants, we will be talking about it in great detail, so do not worry. For now, I hope you are understanding that when there is hypotonic condition outside, which means outside is less concentrated than inside, the water can enter the cell. This water is stored in the vacuole and it maintains the turgor pressure of the cell. When there is hypertonic solution outside, the water leaves the cell first from cytoplasm, then from the vacuole. Okay. Now, second type of vacuoles are called contractile vacuoles. Contractile vacuoles, as the name suggests, are highly extensible. To be able to extend means you can stretch it out. Okay, so they are highly extensible and collapsible. You can stretch it out and you can bring it back to the normal position. Okay, contractile means the same thing, that you can contract it and relax it. Okay. Contractile vacuoles take part in osmoregulation and excretion to maintain the osmolarity and to excrete the waste products. Contractile vacuoles are used. They occur in some protists. Okay, protists are the unicellular eukaryotes. Okay, protista. Remember 5 kingdom classification. So protista are unicellular eukaryotes. Then algae, which live in fresh water. Remember this term, it is important. When the cell, this unicellular organism, are living in fresh water, they will show presence of contractile vacuole. Now what happens? Look at this diagram clearly. You have, because they are living in fresh water is hypotonic. Okay, and these cells may or may not have a cell wall. So what happens is when the water, it keeps entering, okay, this water, excess water coming from outside is concentrated within these contractile vacuole. As more and more water keeps entering, the contractile vacuole becomes larger and larger in size. Okay, it swells. Now this vacuole will move towards the plasma membrane and it will do exocytosis. Okay, when exocytosis happens, whatever water was present inside will be thrown out. So it is like if your boat has a hole, then you take some burton or you take some tumbler and you try and throw the excess water out from the boat, right? So the same thing is happening here, and the tumbler here is the contractile vacuole. Clear? You can see here this is the paramecium, and in the paramecium, okay, it was a video but I think it is not working for some reason. Anyway, it was showing that how water will enter inside these contractile vacuoles. They will grow in size and how it will excrete that water also. Okay, never mind. The third type of vacuoles are food vacuoles. This we already studied when we were studying lysosomes, so it is very easy for you. The food vacuoles are called phagosomes. Okay, and when there is fusion of phagosome and lysosome, then obviously the food will get digested, the nutrients will get absorbed, and the excess will be, whatever the undigested material will be there, will get exocytosed. It is present in protists, several lower animals, and phagocytes of higher animals. Okay, you see it in phagocytes, which are the WBCs, different kinds of WBCs you find in your blood as well as in different organs. Okay, I hope you know this, that apart from your blood, okay, WBC in your blood, organs also have their own WBC. Okay, for hepatocytes they are, which means liver. For liver they are called Kupffer cells. Then for brain they are called microglial cells. Right. So these are nothing but they are phagocytes of organs. Okay. So wherever there are phagocytes in higher animals, these will also have food vacuoles. Protists will have food vacuoles, and some lower animals will show food vacuoles. Okay, their cells will show food vacuoles. Now look here, what is happening is phagocytosis. There is an amoeba. Amoeba extended its pseudopodia, fused it, and now inside you can find two paramecium, okay, trapped. And that, see, they are extending the pseudopodia and trapping the two paramecium, and you can see this specific structure forming. That is nothing but a phagosome. Okay, it is a food vacuole. Fine. This food vacuole you can even see in your WBCs. Okay, this is your white blood cell. It is following the black colored dots, which are the bacteria. Okay, it will keep on following, and then you will see that it will swallow it. Okay, now the bacteria are present inside the cell. Okay, so that inside the cells they are present in form of food vacuoles. The last type of vacuoles are air vacuoles. They are also called gas vacuoles or pseudo vacuoles. Remember this type because it is important. Okay, pseudo means false, nakli. Pseudo vacuoles because they do not have a lipid membrane. Okay, how all the vacuoles have a lipid bilayer, right? A normal membrane. Gas vacuoles or air vacuoles do not have a membrane. Okay, it consists of smaller submicroscopic vesicles. Each vesicle is surrounded by protein membrane, okay, and contains metabolic gases. Now these gases could be anything. If it is it requires oxygen, okay, if it is an aerobic bacteria, it will have oxygen, small bubbles of oxygen covered by a protein membrane. It can also be methane in case of methanogens. Okay, these gases can be different, and they are called metabolic gases because they are either required for metabolism or they are secreted, okay, as byproduct of the metabolism. These air vacuoles are seen only in prokaryotes, only in bacteria. And why? Because they provide buoyancy to the cell. Buoyancy matlab it will be able to float, okay, because of the air vacuoles present inside the cell will be able to float in water. Okay, so for buoyancy, they have air vacuoles. And here we end today's lecture. I hope you had fun and you are solving problems. See you next time.